Hello, everyone. My name is Lauren Sanders, and I am the project scientist for the NASA Open Science Data Repository. This is the second and final lecture of the GL4U RNAseq module set. If you are listening to this lecture, that means you have listened to all three parts of the GL4U RNAseq overview lecture, and you have completed the RNAseq processing Jupyter Notebook hands-on activity. If you have not completed those items, please go back and do so before continuing to listen to this lecture. This lecture is entitled Introduction to Statistics for Differential Gene Expression Analysis. This lecture will include 14 topics, starting with mean, variance, and standard deviation, which are metrics to quantify or describe data. These are going to form the basis for more complex calculations, and in bioinformatics, these are usually calculated by computational programs, although we will learn how to do them by hand as well. We're then going to move on to fold change, hypothesis testing, null hypothesis p-value, adjusted p-value, t-tests and wall tests, principal component analysis, which are commonly used concepts and calculations in probability and significance, which build upon the first three metrics. Finally, we're going to focus on three advanced topics, which are the key methods used in the DESeq2R package for differential gene expression, which is what you're going to be using in the upcoming Jupyter Notebook. These are advanced topics in probability and statistics. And then we're also going to learn a few different visualization and analysis techniques once you have your differentially expressed genes. So some important terminology for the upcoming lecture, and all of these terms are about to be illustrated concretely with examples. But just to start off with, we're going to be using the term sample data or data set interchangeably. This is a set of data points that comes from one experiment. A sample within a data set is a self-contained measurement of one or more variables. And then a data point is a single value in a data set. A variable is a quantifiable aspect of each sample. So this can be numeric or it can be non-numeric. Now, since we're eventually going to be working with spaceflight data, let's imagine that an advanced carbon-based life form named Mel sets out from a planet in the Tadpole galaxy, which is about 420 million light years from Earth. And upon arriving on Earth, Mel accidentally lands in a large field in the iris capital of the world, which happens to be Portland, Oregon. Now, looking around, all there is to see is irises, and so Mel thinks that they are the dominant life form on planet Earth. So Mel spends a year collecting detailed data on petal length and width and sepal length and width of all of the irises. And after doing so, Mel decides that there are three main iris species and names them Setosa, versicolor, virginica. Mel then uses all of this data to create a data set called the iris data set. And if any of you have done any programming in R before or any statistics, you will be familiar with this data set because it's used in a lot of the examples. So Mel goes ahead and sends this iris data set back to the home planet, but the scientists back in the Tadpole galaxy have a lot of questions. So what I'm showing here are the first 5 samples of Mel's data set. And this data set is now publicly available in the R database. So this data contains numerical values for each of the irises for sepal length. So the sepal is the part of the flower which supports and protects the petals. So sepal length and width, and petal length and width, as well as the species that Mel assigned. So going back to that important terminology in the context of the iris data set, the whole data set, we're just looking at the first 5 samples here, but this is called the data set or sample data. One sample is shown in a column here. So in this data set, samples are in columns. And variables are in rows. So one variable, the sepal width, is shown here. And one data point, for example, the petal width of sample 5 is 0.2. So taking a look at this data set, it's just too much to comprehend all at once. So the tadpole scientists want some basic descriptors just to give them an idea of this new species on planet Earth. So Mel starts coming up with different ways to quantify or describe this data. The first quantification that Mel comes up with is the average or the mean of a set of numbers. So to calculate the mean of a set of data points, Mel decides you add all the data points together and then you divide the sum by the total number of data points. And the mathematical equation for the mean is written in this way, where M stands for the mean. The Greek capital sigma stands for sum. X stands for a data point. And then N is the total number of points in the set. We can calculate the mean sepal length for samples 1 through 5 by hand by adding up all of those numbers and dividing by the total number of data points. Thankfully, we're not going to have to do any of these calculations by hand because all of these functions are built into R, so we can do them programmatically. So in R, there's a function called mean, and you pass in a list of numbers. The list you want to take the, and you get the same thing that we would have gotten by hand. Now, Mel then realizes that the mean only describes what the center of the data set is, not the outer edges. And the edge cases, especially in biology, are often the most interesting. So Mel invents another quantification method called the variance or the dispersion of a set of numbers. So one way of thinking about variance is that it tells you the consistency of the points in a data set. So a data set with low variance is going to have data points closer together. It's going to be more consistent. A data set with high variance is going to have data points spread farther apart. And so knowing the variance helps you predict the likelihood of unusual events. If your data set's really consistent, the likelihood of unusual events goes down. Now, what's interesting and why variance adds something to the picture is data sets can actually have the same mean but different variance. So that's why it's important to use both mean and variance to describe a data set. So here we're looking at a hypothetical data set. The data values are on the x-axis. So this particular data set has values from 75 to 125. And the number of samples is on the y-axis, and the mean is 100. Now looking at a different data set, the mean is still 100, but the variance is much greater. The data values span all the way from 50 to 150. So in order to calculate variance, one of the things to keep in mind here is that the way that we calculate variance depends on whether we are calculating variance for the whole population, like all of the irises on Earth, or just a sample out of that population. So Mel's irises data set just came from a field in Portland, Oregon. It's not all of the irises on Earth. Now, the tadpole scientists want to know the variance for the entire iris population, just like biologists want to know everything about whatever organism or species it is that they're studying. But because in this case there's only one Mel and time is short, or in other cases you often can't sample the entire population of whatever it is that you're studying. We and Mel tend to sample what we call a reasonable amount. And then we hope that that sample is big enough to be representative of the entire population. There is a part of statistics that deals with how big a sample needs to be in order to be representative of the population, but we're not going to be going into that today. So in order to calculate the variance of a data set that was sampled from a larger population, we call this the sample variance. And there are a few steps here. So first we calculate the mean of the data set. We then subtract the mean from each data point, square that result, sum up the squared results, and divide by one fewer than the total number of data points. So the equation for variance looks like this, where V is variance. Again, capital Greek sigma is a sum. We're summing up the data point minus the mean, squaring that result, and summing it up, and then dividing by one fewer than the total number of data points. So if we wanted to find the variance of the sepal length for samples 1 through 5, so the sample variance here would be the variance of these 5 irises, whereas the population variance would be the variance of all the irises in the world. So again, we can do this by hand. We can calculate the mean, that's the first starting point, subtract the mean from each data point, square each of those results, sum the results, and divide by 1 fewer than the total number of data points. And then we get 0.046. What do we do with that? Again, we can calculate this programmatically, and we get a slightly different answer just because of rounding is different when you do it by hand versus programmatically in R. But again, this number is a little bit meaningless. 0.046, 0.043, what exactly does that mean to us? Well, standalone, the variance has limited utility, but we can use it to calculate the standard deviation of the data set, which is a little bit more interpretable. So if you take the square root of the variance, the resulting value is going to help us describe the average distance that each data point lies away from the mean. This new metric is called the standard deviation. It's very simple. It's just the square root of the variance. And the reason that this is useful is because it is a measure of how spread out your data points are. We abbreviate standard deviation to SD, or we use the Greek letter sigma, little sigma. So if we take a look at this beautiful fake data distribution, we can see why standard deviation is so useful. So here, mu in the middle stands for the mean, Greek letter little mu, and Greek letter little sigma stands for the standard deviation. So here is the mean. On either side of it, we see 1 standard deviation away from the mean on either side. We could say, okay, 68% of this fake data set is within 1 standard deviation away from the mean. 95% is within 2 standard deviations, and then 99.7% of the data is within 3 standard deviations of the mean. So we can use descriptors like this to tell us how spread out our data is, and we can talk about data points in terms of how many standard deviations away from the mean they are. So the less typical a data point is, the more standard deviations it is away from the mean. So to put this into practice, we can find the standard deviation of the sepal length for iris samples 1 through 5. We can take the square root of the variance that we just calculated. That's easy, 0.21 by hand or 0.207, etc., using the SD function in R. And to put this into practice a little bit more, let's look at the distribution of all of the sepal length data to kind of see how standard deviation is useful. So for iris sepal length, or the entire iris data set, the mean sepal length is 5.84, so that's right here in the middle. And the standard deviation is 0.82. So you can see some of the data is 1 standard deviation away from the mean in either direction, or 2 standard deviations away from the mean, or a little bit more in either direction. Now, this is a histogram plot, which shows the length, sepal length in centimeters on the x-axis, and the number of samples in each of these bars on the y-axis. And the data are colored by the species of iris. So we can now start to ask some questions about these different species in terms of sepal length. So first of all, is the standard deviation of all of the data smaller or greater than the standard deviation of the first 5 samples? So remember, the standard deviation of the first 5 samples was 0.21 or 0.207. So the standard deviation of all of the data is 0.82. So it's greater because, as you would expect, the full data set has more spread out data points than just the first 5. So let's say we wanted to quantify the difference between let's say the Setosa species and the other two species. We can kind of see that Setosa is different because all of the Setosa sepal length data is kind of over on the left-hand side there. The Setosa flowers tend to have shorter sepal lengths. Overall, we can kind of see it visually, but we need to quantify it. We need to put a number on it. So how different is Setosa from the versicolor or the virginica flowers? So let's take a look at the data between the mean and the mean minus 1 standard deviation. Do we see a species-specific pattern here? What about the data between the mean and the mean minus 2 standard deviations? Well, as we can see, looking at the pink pattern here, as we saw before, the Setosa species makes up the majority of the samples between the mean and the mean minus 1 standard deviation, and almost all of the samples between the mean and mean minus 2 standard deviation. So then let's say we got rid of all the Setosa samples. Would the mean of our data set increase or would it decrease? Well, if we got rid of all of those samples, then our distribution would start to be just the blue and the green samples, so our mean would increase. It would fall more between 6 and 7 centimeters. And so then based on this graph, does one of the iris species appear to be more different than the other two? Yes, we can see Setosa has less overlap with the other two than they do with each other. So kind of taking all of this together, what we can see is that we can use the mean and the standard deviation together to not only start to describe a data set, but to describe the different parts of a data set. So how different one species or one group is from the others based on how many standard deviations those data points are from the mean. Now thinking about other ways that you can start quantifying differences between groups in a data set, there's a new metric that Mel discovers called fold change. And so the way that we calculate fold change is we simply take the ratio of the one data point X over the other data point Y. So let's say we wanted to calculate what the fold change is between the data point 6 and 10. 6 divided by 10 is 0.6. So the reason that this is useful is because if the two values were exactly the same, so 10 over 10, we would expect the value of the ratio to equal 1. So a fold change of 1 means no change. But the more different the two values are, the smaller the fold change value gets as it moves away from 1 closer to 0. So you can use fold change as a quick metric to quantify how different or how similar two data points are to, for example, the mean of two groups of samples. So in bioinformatics, we're usually interested in not just knowing the fold change of two data points, but of knowing the fold change of some measured variable, whatever it is, between two groups or two data sets. So in that case, we calculate the mean, the average of our variable of interest for each group, and then we take the fold change of the mean values. So let's say we wanted to quantify how different sepal length is between all the iris species. First, we would calculate the mean sepal length between all of the iris species. Here we're nesting a few functions to get a list from a row in a data frame, sepal length. We calculate the mean of that entire row. We do the same, we do it first for the Setosa species, select out the Setosa species using that row. We do it for versicolor and we do it for virginica as well. As you can see, the mean sepal length is going up as we go from Setosa all the way to virginica, which makes sense based on that graph we were just looking at. And then if we want to calculate the full change of sepal length between all the iris species, all we have to do is take those ratios. So Setosa over versicolor is 0.84, Setosa over virginica is 0.76, and versicolor over virginica is 0.9. So based on the fold change values, which of these iris species are the most similar? So remember that a fold change of 1 means that the values were exactly the same. As fold change gets closer to 0, the points were more and more different. So the two iris species that were the most similar would be versicolor and virginica. And then the two that are the most different would be setosa and virginica, which makes sense based on that plot we were just looking at, where setosa was all the way on the left-hand side and virginica was on the right in terms of sepal length. Now, in practice, if we're looking at a whole bunch of variables, like for example, thousands of genes, which is what we're going to be looking at soon, the values for fold change can be greatly spaced out. Like they're not going to be, it can be sometimes like 0.9 all the way down to 0.00001. So this makes it difficult to compare and visualize them directly. So we will often take the log of the fold change values. And so we report this as log fold change or LFC. So in this case, we again can do this in R, where we take the log of our ratios. The setosa over versicolor log fold change is 0.17, and it's just the inverse for versicolor over setosa. So this is really easy to interpret because we know that a negative log fold change indicates lower overall values in the group being compared. So it's the numerator, that's setosa. Whereas the inverse, a positive log fold change, indicates higher overall values in the group being compared. So it's more interpretable. It's easy to quickly understand what's going on. So taking a look at the iris data set again, what has Mel been ignoring so far? Well, we've only really used this one row, this one variable, sepal length. We also used the species variable. We haven't used any of these other numerical variables. This is what we call multidimensional data. So it has more than one variable, where when we say multidimensional, one dimension in that phrase means one variable. So how can Mel start to work with all of these different variables, not one at a time, but merge them, combine them all together to more completely analyze the differences between the three iris species? So not just sepal length, but how do these species differ in terms of sepal width, petal length, petal width, kind of paint a more complete picture of what's going on. So one thing that Mel can do is to create a 2D scatter plot. So this will paint a picture of the relationship between two variables. So here we have a 2D scatter plot where we have sepal width on the y-axis and sepal length on the x-axis. And we've colored the different dots in the scatter plot by the different species. So here we can see how sepal width compares to sepal length, and then how the different species compare to each other. But here again, Mel is only looking at two variables, okay? We can even make a 3D scatter plot. It's a little harder to visualize, but here's a 3D scatter plot where you can see petal length on the y-axis, sepal length on the x-axis, and then sepal width on the z-axis, okay? You kind of need an interactive visualization here so that you can kind of turn it around and look at it from all dimensions. But there are 4 variables here, and even with very advanced technology, it is difficult to visualize 4 dimensions. So Mel gets super ambitious and invents principal component analysis, or PCA. So PCA is a very commonly used bioinformatics technique for high dimensional, multi-dimensional data sets. In our case, we're going to be working with gene expression data where each variable is a gene. There are thousands of variables. And PCA is useful because it reduces the number of dimensions or variables by condensing all of these variables into a small number of vectors. So another way of saying this is PCA is a dimensionality reduction technique. So here when we say small number of vectors, a vector is a summary of the information. So a summary of the variability, the variance of the data set, which was previously spread out across all these different variables. So the vectors represent the main sources of information or the main sources of variance, the main reasons why variance is being introduced into the data set. And these vectors are called principal components. We often abbreviate this to PCs. And high dimensionality data, like gene expression data, are usually visualized using a scatter plot of the first two principal components. So we kind of assume the first two principal components will usually represent a majority of the variance in the data set. And I'll show you what that means in a second. But first, how are principal components calculated? Well, first, we calculate the covariance matrix between all data points. So if variance is how a data point varies from the mean, covariance is how similarly two data points vary from the mean. So we're comparing pairwise, we're comparing all pairs of data points in the data set. And we're saying how similar is the variance between each pair of data points. So we're laying out this relational matrix that tells us the relationship between each pair of data points in terms of how similar each one is to the mean, or how far away it is from the mean. So the question that we're asking is, what are the relationship between each of the pairs of points? What's the correlation between each of the pairs of points? We then calculate what's called the eigenvectors of the covariance matrix. So this is getting into some advanced math. We're not going to get into the details, but you can think about eigenvectors as the directions within the covariance matrix where there is the most variance. So we have two data points being compared all over the matrix. Where's the most variance? Where are the data points most different from each other? Because that's really what we're interested in the data set. If data points are really, really similar to each other, that's not as interesting. So once we have these eigenvectors, so where's there the most variance, these are the principal components. So this is a visualization showing how principal components are calculated. So if you take a look here, while the spinning axis is aligned with these two purple lines, the first principal component within this is a scatter plot of, let's say this is all the data in our data set. We can see that the most variance within the data set, the most change within the data set is happening from the lower left to the top right, so along these purple lines. So that's going to be our first principal component. So when this long axis is lined up with the purple, that's the first principal component. But then you can see there's also some variance within this data set going in the opposite direction. So that the dots are also spread out going from the bottom right up to the top left. There is some spread, not as much as going from the bottom left to the top right, but there is some spread going from the bottom right up to the top left. So this is called the orthogonal, or the perpendicular direction to the first principal component. So if the first principal component is along the purple, the second principal component is always going to have to be this perpendicular line. It's going to be exactly perpendicular to that first principal component to try to capture most of the remaining spread, most of the remaining variance or difference within the data set. There are other axes here. As you can see, there is other spread in pretty much every other direction. You could say, oh yes, the data is spread out this way, it's spread out this way, it's spread out this way. But if we take the first principal component going in this direction, we take the second principal component going in the orthogonal or perpendicular direction, we're capturing most of the variance within the data set. And so that's a way of taking all of these variables, all of these data, not data points, but variables that were measured for data points, and capturing all of the variance, most of the variance along the first principal component and the second principal component. So it's a way of reducing down this dimensionality just to measurements. So now if we were to take a look at a PCA plot of all 4 iris variables condensed into 2 principal components. So here, the first principal component, which captures 72.96% of the variance, so pretty good, a lot of variance in the data set. The first principal component is along the x-axis, and the second principal component, which captures 22.85%, is along the y-axis. So this is now a scatter plot where each of the points is an iris sample. And instead of the X and Y axis being things that were actually measured in the data set, like sepal length or sepal width, now each data point has two values. First principal component, the second principal component, where the value represents where this data point falls within the variables that were contributing the most to principal component 1 and principal component 2. So the variables that are causing that spread along the principal component 1 axis or the principal component 2 axis. So if we take a look at how much variance within the data set is explained by this plot, we have, 72, 73 approximately plus 23 approximately, which is about 96%. So this principal component analysis was very useful. It reduced our dimensionality from 4 variables down to 2 and captures nearly almost all of the variance within the data set. So this is useful. And what we can see is each of these dots is one sample, and these are the percent of data set variance that were captured. What we can see is PCA is very useful because it's able to, once we perform a scatter plot of all the data points using the 2 principal components, PCA automatically finds these clusters of similar samples. So all the setosis samples are over here on the left. They have really low PC1 values, whereas cluster 2 has the versicolor and virginica samples all together here. So PCA is very useful because it finds these relationships within samples. So Mel, who happens to know about DNA, now thinks that the differences that are being observed between the iris species are probably due to a gene that's turned on in the setosa flowers, but off in the versicolor and virginica flowers. So what would be the name for this type of statement? So we call this type of statement in science a hypothesis. And we can then test this hypothesis. So a hypothesis is just an educated guess based on some data that you have. So here Mel's looking at all the iris data and saying, I wonder what could explain this clustering pattern. Maybe it's a gene that's turned on and off. Now, a hypothesis has to be testable in some way, otherwise it's useless. So let's say we come up with a hypothesis based on everything that we've seen. Okay, all sheep are white. Well, lots of white sheep support this hypothesis, but only one purple sheep is enough to disprove it. So this hypothesis is testable. If you see a sheep that's not white, there you go. Now, in science, we rely a lot on what we call the null hypothesis. So the null hypothesis is the current or the existing explanation. And in science in particular, we will often say the null hypothesis is, okay, the pattern we're observing is just coincidence. It's not actually due to some biological phenomenon. So we say in order to accept our hypothesis, if we can't, sometimes we can't directly test it, so we must reject the null hypothesis. But if we usually can't decisively disprove the null hypothesis, we can't prove that this is never just coincidence, how do we ever reject it? Well, first we design and perform experiments that will collect data that either supports our hypothesis or not. And then we can perform statistical analysis on the data we've collected to see whether our results occurred by chance. That's what statistics are for. And if we can demonstrate that our results likely did not occur by chance, then we can reject the null hypothesis. So what would the null hypothesis be for the hypothesis that Mel put forth, which is I hypothesize that the differences I'm observing between all of these species are due to a gene that's turned off in setosa, but off in on in setosa, but off in the other species. So the null hypothesis would just be the differences observed between all of these species are not attributable to a gene turned on in setosa, but off in the other species. And what would be a good experiment that Mel could perform to test this hypothesis? Well, we just learned about RNA sequencing, right, which quantifies the level of gene expression in samples. So what Mel could do would be to extract RNA from several replicates of each iris species and perform RNA sequencing. Now, getting into how we would use statistics to test whether our results occurred by chance, one concept that is really important for hypothesis testing is the p-value or probability value. So the p-value is the probability that your null hypothesis is true. So given the data that you get from your experiment, what is the probability that either everything occurred by chance or everything is not attributable to whatever you thought it was attributable to? So in this case, the lower the p-value, the less likely the null hypothesis is to be true. And we generally will accept a p-value of less than 0.05 as low enough to reject the null hypothesis. So here we would say there's only 5% chance that the null hypothesis is true. And then if our p-value is less than 0.05, we can accept our alternate hypothesis. So p-value of 0.05, another way to put this is this indicates statistical significance. So the outcome of our experiment was likely not due to random chance. So there are several different statistical hypothesis testing methods to calculate p-values, and we're going to discuss two of these in the next few slides. We're going to discuss the t-test and the Wald test. Now, given what we've just discussed, is it ever possible to definitively prove a hypothesis using hypothesis testing? And the answer to this would be no. The best we can do is to assign a probability to the null hypothesis being true. So all we can do is say there's a very, very, very low probability that the null hypothesis is true. There's a low probability that our result occurred by chance. Therefore, a high probability that whatever hypothesis we were testing using our experiments is true. Now, all of this depends on us actually designing good experiments that do directly test our hypothesis. But assuming that that was true, all we can do is assign a probability to the null hypothesis. We're never actually proving a hypothesis, and that's very important. Now, another concept that's going to be important when we start looking at our gene expression data is the concept of adjusted p-value. So let's say Mel went ahead and performed RNA sequencing on the irises. Well, some iris genomes have over 100,000 genes. In this case, each gene is a variable. This is what we're measuring in our experiment. So RNA sequencing would give over 100,000 variables to compare between the iris species. When we get into this situation where we have data with thousands of variables, we need to mathematically adjust our p-values for multiple comparison testing in order to avoid false positives. So what this means is multiple comparison testing is if we are comparing multiple variables between conditions, we're not just comparing one or a handful, and we'll get into how many you need to be testing to get to the point where you have to adjust for multiple comparison testing. But if we're comparing multiple variables, like thousands of variables, thousands of genes between conditions, so in this case, between different iris species, we need to adjust our p-values. So why is this important? Let's say Mel was just looking at 20 genes. We'll back up for a second. Let's say where there were just 20 genes. If Mel's looking at the difference in expression of a random set of 20 genes, just by chance, we would expect that on average, 1 out of 20 genes is going to appear significant if we use the 0.05 or 5% p-value cutoff. This is what we call a false positive. So this is a result that appears significant, but in reality, this is due to random chance. So mathematical p-value adjustment takes into account the probability of false positives given the number of variables. So many bioinformatic tools are going to generate a p-value, which doesn't take these false positives into account, and an adjusted p-value, which does. So we're not going to get into the actual math behind this, but all of the p-values are mathematically adjusted and generally increased in value in order to take the amount of variables that were being tested into account. So just to double check knowledge here, what should be used when we're determining the significance of an experiment where about 100,000 genes were considered? Should we use p-value or adjusted p-value, and why? Well, we're going to want to use adjusted p-value because it takes into account the likelihood of false positives. And then when is it appropriate to use a p-value rather than an adjusted p-value to determine significance, given that we're using the generally accepted p-value cutoff for significance of 0.05 or 1/20? Well, if you've designed an experiment where there are few enough variables that false positives are not reasonably expected. So if you're using a p-value cutoff of 0.05, so 1/20, you would want far fewer variables than 20 so that by chance you're not expecting to get a p-value low enough just by false positive. But then this is important. Which value is going to give you more significantly differentially expressed genes in Mel's experiment, the p-value or the adjusted p-value? Well, because adjusted p-value increases all of those p-values to take into account the possibility of false positives, the unadjusted p-value is going to give you more significantly differentially expressed genes because it's a less stringent metric. So how do we actually calculate a p-value? So in order to calculate a p-value, we're going to use a statistical hypothesis testing method, such as a t-test. So a t-test compares the means of all measured variables between two groups. So let's say we're looking at the expression of gene X, that's one of our measured variables, in two different species of iris. We're going to look at the mean of the expression of that gene in those two different groups. And then in order to calculate the t-score, which is what comes out of a t-test, we're going to look at what we call the signal versus the noise, or the difference in the group means versus the variability of the groups, how spread out those groups are. So the way that we do that is we take the mean expression of that gene in the one group minus the other group over the standard deviation of that gene in the one group versus the other group. And we can see, so standard deviation is coming in handy again to help us describe how different these two groups are using both mean and standard deviation. So the results returned from a t-test are a t-score or t-value for each variable, where the greater the t-score, the more difference there is within that variable between the two groups. And then there's a p-value associated with each t-value. And so this p-value is describing the likelihood of the null hypothesis being true for that variable. Now, another hypothesis testing method is the walled test, and this is what's going to be used in DESeq2 to test the null hypothesis that is this gene differentially expressed between two groups. So the way the walled test works is it creates a mathematical model for the null hypothesis and our testing hypothesis, where here our testing hypothesis is this gene is differentially expressed between these two groups, and then identifies which model best maximizes the likelihood of the data that's actually being seen. So mathematical model here just means a mathematical description of a situation. So specifying rules about how data should behave within that situation. So this is what the data should look like if this gene is differentially expressed between two groups. So we can put this another way. The wald test identifies a mathematical model for which the data that we have is the most likely result. So the wald test returns a wald statistic for each variable, and then a p-value, again, associated with each wald statistic. So the wald statistic is a lot like a t-score. It's a value that describes whether we can reject the null hypothesis for that variable, and it's useful for ranking genes for downstream applications. So we're going to use it to rank genes for gene set enrichment analysis. So this is a method that identifies gene sets that uses a ranked gene list. There's another method that we're not going to use, but is useful to know about, called ingenuity pathway analysis, or IPA, which finds biological pathways enriched within a gene data set. So going over some questions about these statistical testing methods. If MEL's final gene expression data has measurements from 100,000 genes and a t-test is performed to compare the two groups, how many t-values and p-values are expected? Well, since we are testing 100,000 genes and each gene is a variable, we're testing that same hypothesis for each gene. We're testing the hypothesis that that gene is differentially expressed between two groups. We would expect 100,000 t-values and 100,000 p-values. What if MEL performs a wald test? Same thing. We would expect 100,000 of each values. Now, getting a little bit more in depth into the methods that we're going to use for RNA sequencing data. So it's important that we normalize our RNA sequencing data for differences in read depth. This is because gene expression data often have different read depth levels per sample, where read depth is the total number of RNA reads that come off the sequencer for each sample. This is because the number of reads in a sequencing run is split up between the number of samples. So the read depth for each sample just depends on the total number of samples that happened to be sequenced that day. So you can get into a situation where you're comparing samples that have different numbers of reads simply because of a sequencing artifact. So if MEL directly compares these two samples that we're looking at in this graphic, it's going to look like all genes are more highly expressed in sample 1. So MEL first has to normalize all of these samples by their read depth in order to allow direct comparison of gene expression values between samples. So when we use DE-seq2 to perform differential expression analysis, DE-seq2 performs normalization across all the samples using the median of ratios method, and this gene expression is being normalized by read depth. So this is DE-seq2 step 1, normalization, and for each, the way this happens is that for each gene, a pseudo-reference sample is created that's equal to the geometric mean across all samples. So geometric mean is a little different from the mean we learned about earlier. It's the square root of all the products of all of the data points. So let's say we have a tiny little data set here where we've measured the expression of these two genes for these two samples. A pseudo-reference sample is created that it takes the square root of the product of all the data points. The pseudo-reference sample is then used. A ratio is calculated of the actual sample, sample 1, over the pseudo-reference. This is performed for each sample in the data set. And now we know in biology, the majority of genes are not going to be differentially expressed. Only a few genes will be differentially expressed. And so therefore, the majority of genes in each sample should have similar ratios within the sample. So you can see that, for example, with this ratio where we've calculated sample 1 over this pseudo reference, most of these genes fall within the 1.2, 1.3 range. There is one gene here that's a lot lower, so that's potentially truly differentially expressed. We then take the median value of all the ratios for a given sample, and that's used as the normalization factor or the size factor for that sample. So each sample gets its own normalization factor. And then each raw count value is divided by that sample's normalization factor in order to generate the normalized count values. So, for example, if we look at this gene in the raw counts, ABCD1, you think, well, in sample 1, 22 is nearly twice 13, so this gene must be significantly differentially expressed in sample 1. But after normalization, this gene actually comes out to be very similar between sample 1 and sample 2. Now, we still don't know whether this gene is significantly differentially expressed because we haven't actually done the differential gene expression analysis, but this is an example to show how the normalized samples give more reliable insight. So let's say we didn't perform this read-depth normalization. What would we expect if we compared gene expression values between sample 1, where the read-depth was 5 million reads, and sample 2, where the read-depth was 20 million reads? Well, we would accidentally think that most of the differentially expressed genes would seem like they were way overexpressed in sample 2 because more raw counts are being assigned to each gene in sample 2 because of that deeper sequencing depth that sample 2 got. And this would be problematic because this is not, maybe, but we can't be sure if this is representative of the true biology. And some of this may be due to biology, some of it may be due to sequencing depth difference. So after we've taken care of that sequencing depth issue, we are ready to perform differential expression analysis using DESeq2. So once again, we could think about this as 100,000 hypothesis tests run at once, one for each gene, where we're testing the hypothesis, is this gene differentially expressed? And DESeq2 uses a walled test for the differential gene expression analysis. And like I said, the null hypothesis we're testing for each gene is this gene does not change expression significantly between the tested groups. So for genes with low mean counts, it's important that we estimate gene-wise variance before the walled test. So our variance or dispersion is the word that DESeq2 uses. These estimates are often unreliable. So to solve this problem, DESeq2 estimates variance and dispersion by sharing information across genes. And it's important to have these reliable variance estimates for the walled test. So with this approach, DESeq2 is assuming that genes with similar expression levels have similar dispersion or variance. And the dispersion for each gene or variance is estimated using a technique called maximum likelihood estimation. So this means that the most likely estimate of dispersion is calculated for each gene, given the gene expression values from all the replicates in each group. And finally, DESeq2 step 3, we're using the walled test to generate a mathematical model. So recall that the walled test creates a mathematical model for the null hypothesis and the testing hypothesis and identifies which model best maximizes the likelihood of the data. So the important thing to keep in mind here is that the type of model is dependent on the shape or the distribution of the data. So the distribution shape and the numbers that specify that shape, so the mean and the variance, the spread outness of the data, that's what this model is. So the wall test has to be told, what does the data look like? What type of general type of distribution are we supposed to use here? And we can then generate the mathematical model. So RNA-seq data has a very specific shape. So the majority of the genes have an expression value close to 0, but there's this long right-hand tail because there's no upper limit for maximum expression value. So most of these genes have low, but there are some genes that have really, really high expression. So this count data is often modeled with the binomial distribution. So the binomial distribution is also called the coin toss distribution. It's illustrated by the probability of getting heads when you toss a coin a certain number of times. So in this graph, if you toss a coin 40 times, your probability of getting heads is about half the time, right? But not all count data can be accurately fit with the binomial distribution because this is only used in situations where there's a relatively small number of data points. So instead, we tend to use the negative binomial distribution for RNA sequencing data. So this is the negative binomial distribution. And you can notice this looks really similar to what you would expect for RNA-seq data. So it makes sense that we would use the negative binomial distribution for wald testing. All right, so finally, we're going to get into a few ways of visualizing and analyzing RNA sequencing data, because after differential expression analysis, we may have hundreds or even thousands, if we're lucky, of differentially expressed genes. It's easier to interpret a visualization. And so two common visualizations for RNA sequencing are the heat map and the volcano plot. So heat map, one of my favorite visualizations, is where you take a numerical matrix. Here I've got samples as columns and genes as rows. And you turn it into a matrix where each of the values is preserved in its location in the matrix, but the actual expression value is color coded, where here, this is why it's called a heat map. Red corresponds to high values, and blue corresponds to low values, so kind of like hot and cold. We can also cluster our rows and our columns based on the similarity of values, which can illuminate different patterns within our data set. We do have this legend or color bar where the colors are mapped to this range of values within the matrix. But notice something here. Do you think that all the data values in our matrix naturally fell between -2 and 2? Especially if we're talking about gene expression data where we just said most of the values are close to 0, but there's this long right-hand tail because these values can be very, very large. These data have been scaled so that they fall between -2 and 2. So here's a little illustration of what scaling means. So pre-scaling, gene count values can range from 1 to tens of thousands. And these larger values, which you can see out here as stars before scaling, can skew the data non-representatively. Scaling brings all the values into similar proportions. So you'd see their relation to each other is the same, but all the data values are similarly proportioned, so they can be reasonably compared. Another plot that we can do is a volcano plot. So remember, after a differential expression analysis, we're going to get our p-value, we're going to get our adjusted p-value for all our genes, and we're also going to get a log2 fold change value. So we can then generate a volcano plot. We're going to go through all the aspects of this plot in a moment, but what this will tell us is, at a quick glance, kind of how many genes are differentially expressed up and down. So a positive versus a negative log2 fold change, and with the smallest p-value. So if we take a look at the different aspects of this plot, we have log2 fold change on the x-axis. We have a negative log 10 of the adjusted p-value. And the reason that we take the negative log 10 is because as the adjusted p-value gets smaller and smaller and smaller, it's hard to visualize on a plot. But the negative log adjusted p-value gets bigger. So we also use significance cutoffs in our volcano plot. And on the plot, these cutoffs are shown with dotted lines. So for example, in this case, a gene is colored green if its log two-fold changes between the two compared conditions is greater than absolute value of 2. And a gene is colored blue if its adjusted p-value is less than 10 to the negative 6. A gene is colored red if its log two-fold change is greater than absolute value of 2 and its adjusted p-value is less than 10 to the negative 6. So we use these adjusted lines to create these areas of the plot so that we know just by looking at the plot what the significance of the differential expression of each gene is. So we end up with a plot that looks like this, where again, the most significantly differentially expressed genes are within the red quadrants with the dotted lines. Then we have genes where our adjusted p-value cutoff only is within blue, and genes that only fell within our log two-fold change cutoff are in green, and then genes that were not significant are in gray. So we also have our legend at the top and name labels for some of the significant genes. Finally, the last analysis we're going to be performing is gene set enrichment analysis, also referred to as GSEA. GSEA was developed in a landmark paper in 2005. And from this paper, the mathematical derivation is if genes can be ordered in a ranked list L according to their differential expression between the classes, so you can rank your genes by log two-fold change, you can rank them by adjusted p-value, and then given an a priori defined set of genes S, so this is usually a gene set that defines some biological function, so all the genes that are involved in some molecular pathway, for example. The goal of GSEA is to determine whether the members of S are randomly distributed throughout our ranked list L or found at the top or the bottom. So in this plot from the paper, you can see we have a ranked gene list compared between two classes. We have our gene set S, and if we take a look at our gene set, which again, some molecular biological function, we can see that these genes seem to be clustered more at the top of the ranked gene list. So they're enriched at the top, meaning that we can say, okay, this gene list seems to be activated. This gene set seems to be activated in our gene list. In our analysis, we're going to be using gene ontologies. I'm showing here there are a lot of different collections of different gene sets available on the molecular signatures database. The ontology gene sets are one of them. There are the gene ontology biological process, over 7,000 gene sets, the cellular component gene ontology, and the molecular function gene ontology. So these are all gene sets or gene lists that represent different biological process, cellular component, or molecular function. If we take a look at a few of the gene ontology biological process gene sets, there are things like microRNA metabolic processes, microRNA processing, mismatch repair, etc. So the GSEA results that we're going to be investigating in our notebook, we're going to represent them using a dot plot and an enrichment relationship plot. So in our dot plot, what we're going to do is on each row of the dot plot, we're going to put the different gene sets that were found to be either activated or enriched through, activated or suppressed through enrichment in our ranked list of genes. Each dot on this dot plot, the size of the dot represents the count of genes from each gene set that were differentially expressed, and the color of each dot represents the value of the adjusted p-value. We're also, you can look at the same type of data, but in a relationship plot, which shows if there are some genes that overlap between different pathways, they will be joined together by these gray edges in an edge and node formation. And then gene sets that did not have any genes overlapping with anything are held out on the sides here. So again, the size of the dots correspond to the number of differentially expressed genes within each pathway, and then the color of the dot has to do with the adjusted p-value. So this concludes the lecture portion of the GL4U RNAseq module set. You're now ready to dive into the RNA-seq analysis Jupyter Notebook hands-on activity where you put the concepts you just learned into practice.